We're now going to turn our attention to the focus of membranes and membranes transport and the form and the function of membranes and what's in them and how they actually function to enable cells to carry out all of the reactions that they do. And this is for section B2.1. The cell or plasma membrane is essentially the border between the cell and its external environment. It helps to create an environment inside of the cell, the internal environment, that can be different from the external environment. And the structure of it is the same for all organisms. As we've discussed, it's made up of a bilayer, so it's two layers of phospholipids that are amphipathic molecules, meaning that they are hydrophobic and hydrophilic. In terms of how big it actually is, it's not something that typically you'd be able to see under a light microscope. They're about 10 nanometers or less across in size, so they're quite small. The bilayer is composed of two layers of phospholipids, and this would be an individual phospholipid would include the phosphate head and two fatty acid tails, and it arranges itself so that the tails are protected from aqueous solutions, and it forms an enclosed shape so that the two tails, the portions that are hydrophobic, are facing one another, and the hydrophilic phosphate heads are facing the external and the internal portions of the cell, which would be where the aqueous solutions are at. Again, the function is to control essentially what comes in and out of the cell. It helps to create that internal environment separate from the external. It's also permeable, meaning that it's able, can allow some molecules to pass across the membrane, and as we'll see, some molecules need some assistance to be able to do so. Due to the polarity of the middle of the membrane where the fatty acid tails are at, the membrane itself has low permeability to particles that are hydrophilic, including water and glucose. It's possible for them to pass across the membrane, but it's slow, doesn't happen very frequently or easily. And the external and the internal portions of the membrane we see are aqueous. So inside and outside, and aqueous we mean a water and some composition of solutes. If hydrophilic molecules reach the core of the membrane, so if they get in within those fatty acid tails, oftentimes they're expelled due to polarity attractions. They're attracted to those hydrophilic portions, and so they get expelled out. Typically, the larger the molecule, the less permeability it has. Oxygen is easier to pass through than glycogen or proteins. And so usually larger molecules and molecules that are neutral, don't have positive, negative, or nonpolar, are smaller particles and nonpolar are going to be easier to pass across the membrane. Molecules are in constant motion. They're continually moving around within their environments, and they typically will move to areas of high concentration to low concentration in order to meet a balance point or homeostasis. And this is referred to as simple diffusion. It's the movement of molecules, whether it be liquid or gas, sometimes across a semi-permeable membrane, not necessarily required, until homeostasis or a balance is met. And this does not require any energy because molecules are always moving around, eventually spreads out. A good way to think about this, if you light a fire, the smell of the fire is really strong right where the fire's at. If you're sitting across the room or across the campsite, eventually you can start to smell that. That's because the particles from burning the fire begin to spread or diffuse out, and you can eventually smell it. And so that's the movement of those molecules moving around and moving from areas of high concentration where the fire's at to lower concentration where you're sitting across the room or across the area. And so this is a net movement of from areas of high concentration to low concentration. Diffusion across the membrane is possible because the membrane is permeable to some particles. Oxygen, again, is a good example because it's nonpolar. The center of the membrane, as we've discussed, is hydrophobic, so ions can't easily diffuse across it. Polar molecules can diffuse at really low rates. And again, the smaller the molecule, the easier the rate of diffusion or how quickly it can take place. Urea and ethanol would be examples that are able to move across the membrane. As discussed with proteins, one of the functions of proteins is transport, and there are proteins embedded within the membrane that assist sometimes with helping to move molecules across. And there's two main types of proteins within the membrane: integral proteins and peripheral proteins. Integral proteins are those that fit across at least one layer or pass across the entirety of the membrane. An example of these would be like a tunnel or a channel protein. They are hydrophobic on at least one part of the surface. And they're often transmembrane with hydrophilic external regions, so on the outsides in the aqueous solutions, and hydrophobic internal regions where those fatty acid tails are at that are hydrophobic. Peripheral proteins, on the other hand, are on one side of the membrane, either internal or external. They don't cross the entirety of the membrane. They're hydrophilic on one surface, so sometimes they're not embedded within the membrane. Some have hydrocarbon chains that help to anchor those proteins to the membrane surface so that they don't move around and that they're located and stuck on that membrane, on the membrane. These integral and peripheral proteins can have a number of different functions. For example, look at a couple of examples here. They can act as a hormone binding site. The protein receptor has a binding site with a specific signal molecule, a hormone that it will bind to, and it's specific to that particular type of hormone, so other hormones are not going to be able to bond typically. And the binding of that hormone causes the protein to change shape, and it releases some chemical signal. So the hormone comes, it binds to the protein, and then it releases some signal within the cell that then causes something to happen. An example would be the hormone epidermal growth factor, EFG, and it binds to an integral protein to send signals. And a second example would be an immobilized enzyme, meaning that it's stuck there, it can't move around. And this is a protein that's built into the membrane, and it serves as a catalyst to carry out different functions. And sometimes it may be multiple enzymes working together in a series or in a chain. We'll see some examples of these in the production of ATP through the electron transport chain, and a specific single example would be ATP synthase, the enzyme that actually produces or couples ADP with phosphates to produce ATP. A third example would be cell adhesion, so actually holding cells together. Membrane proteins can be used to connect to another cell's membrane as well, to other proteins within a different cell's membrane, and so it can form what's called gap junctions or tight junctions, and can hold those cells together, helps to produce can help to produce tissues. And lastly, cell-to-cell communication. Cells communicate with one another through chemical signaling, and glycoproteins, so proteins that have the carbohydrate chain on them, can serve as indicators for other cells. They can also sometimes serve as short-term bonding, and we see these as examples within nerve cells. One of the essential molecules to life, as we discussed at the very beginning of the course, is water, and water moves in and out of cells continuously. There's no net movement, and so this would create a balance or homeostasis, so water's moving in and out continuously unless the concentrations in or out of the cell change of solutes, but typically it's just a continual movement in and out, maintaining homeostasis, and this net movement causes osmosis, which is the movement of water molecules across a selectively permeable membrane. Osmosis is a type of diffusion, and one of the primary differences or distinguishing factors is that osmosis is specifically with water. And this occurs because of differences in concentration of substances that are dissolved in the solute. And so the solvent is the item that's being dissolved. The solute is the thing that is dissolved. Solvent is doing the dissolving. And so typically what we see is areas of higher concentration of solute have a lower concentration of water molecules due to the bonding of the water and the solute restricting the water movement. And so it's a net movement of water from where there's low solute concentration to regions where there's higher solute concentration. I like to think of this as kind of the opposite. Essentially, it's still water molecules moving from where there's lots to the little, high to low concentration. That's for water specifically, or the solvent. For osmosis, it's the low solute concentration to regions of higher solute concentration. The water is moving in that direction. Osmosis is an example of passive transport. It does not require energy, and it occurs in cells as water is small enough to move across the bilayer, the membrane, even though they are hydrophilic, the water molecules. Sometimes a particular protein called an aquaporin can help to move molecules, water molecules, across the membrane. It's a channel transmembrane protein, and it's just slightly wider than water, and so they must pass single file. Hydrogen ions and other small molecules can't use that aquaporin, hydrogen ions specifically because of their positive charge. And so that channel protein is specific to water. Although some molecules can pass across the membrane through simple diffusion, most molecules need some assistance. And an example of this would come through channel proteins, just like the aquaporins that we looked at. And this would be called facilitated diffusion. There's something that's helping to facilitate the diffusion, still from high to low concentration. And what's doing the facilitating is an integral protein, a transmembrane protein with a pore that connects the cytoplasm to aqueous solutions inside or outside of the cell. The pore diameter, so how big it is, and the chemical properties of it ensure that only a particular type of molecule can pass through. Sodium or potassium ions for a transmembrane protein, but not both. It's one or the other in this example. And these are really helpful in assisting ions and polar molecules to move across the cell membrane because they wouldn't otherwise be able to. And so it allows also movement in and out of the cell, but it's still high to low concentrations. It's still an example of passive transport. No energy is required. And molecules move across or along with the concentration gradient. And so this is the idea that molecules are continually moving, but they move from areas of high concentration to low concentration to reach that homeostasis, that balance. And so then this represents a type of diffusion. It's just that the protein is helping to facilitate it. It could be a channel protein, a tunnel protein. Also sometimes proteins will be gates and open and close, but always still passive transport, no energy required. Sometimes the cells can actually open and close the channels to control the permeability, so allowing things in or out at specific times. And a good example of this would be glucose and amino acid transport. Both require facilitated diffusion due to their size. They're larger molecules than what we would see oxygen, for example. For passive transport, we've looked at simple diffusion and facilitated diffusion, both requiring no energy. And active transport is the opposite. And this would be, for example, pump proteins or protein pumps that are used to move molecules against the concentration gradient. So they're moving from areas of low to high concentration. This requires energy, and I like to think of it as trying to pump water uphill. It would require energy or effort to be able to make that happen, and that energy comes in the form of ATP. And so it's moving molecules in one direction against the concentration gradient. The pump changes its conformation shape in order to be able to move the particular molecule to the opposite side. Again, ATP would be required for this to occur. After that movement, though, the pump then returns to its conformation, and that does not require additional energy. So it'd be like pumping something uphill and depositing energy to pump it uphill, but that water could rush back downhill. Wouldn't require additional energy. It's kind of an analogy. A good example of this that we'll discuss a little bit further would be sodium-potassium pump, in which sodium is pumped out of the cell and potassium into the cell, and that happens both against their concentration gradients and is assisting nerve signals. Cell membranes are permeable, and they can be permeable in a couple of different ways. Semi-permeable would be the ability or the allowing of passage of some small particles, and also freely permeable to the solvent. Simple diffusion follows this in that it depends on the size and the polarity of the particles being able to move across the membrane. Selectively permeable allows passage for particular molecules, and facilitated diffusion and active transport would allow this because it depends, again, on the size and the polarity of the protein, but there's something that's facilitating the movement across the membrane, and that protein, that transmembrane protein, must match the particular particle, the solute, whatever is moving across. And so it's based off of the size and the polarity, but it's unique. It's specific to that molecule. It's exclusive to that particular molecule that's being moved across the membrane. Within the membrane, we can find both glycoproteins and glycolipids. And glycoproteins are conjugated proteins that have a carbohydrate connected to the protein. They're typically partially embedded within the membrane and partially exposed to the extracellular matrix. the space outside of the cell, and typically that's going to be the carbohydrate portion. As I mentioned previously, they assist in cell-to-cell communication and recognition, and they also allow the immune system to distinguish between self and non-self. The other example would be glycolipids, and these are carbohydrates that are linked to lipids. The carb is usually a single monosaccharide or a short chain of 2 and 4 sugar units. The lipid is 1 or 2 hydrocarbon chains that fit into the hydrophobic core of the membrane, so it's down in those fatty acid tails. It occurs in eukaryotes with the carb portion pointing out towards the extracellular matrix. Together, they form the carbohydrate outer layer with aqueous solution between the gaps between the different cells, and this forms the glycocalyx, which can fuse between adjacent cells and helps to bind or keep that tissue together, helps to form tissue through the binding of those cells. Our current evidence supports the model of the membrane called the fluid mosaic model. And this is the idea that the membrane is fluid. The plasma membrane is able to bend and flex and change shapes, much like water. It's a mosaic because it's composed of multiple different types of molecules. We have phospholipids, proteins, cholesterol, carbohydrates. All of these different components make up a mosaic of the membrane, kind of like a mosaic picture or painting. And it's also a model. It's a representation for a real item. And so this is what we call or can describe the membrane as. Those phospholipids can move freely. They have weak attractions, but combined together, they create a strong membrane that's able to bend and flex, representing the fluid mosaic model.